Hello, good morning, everyone. Let's get started. We will, as usual, review a little bit of what we did last week, last class, before we continue from where we left off. So please take out your study guide if you have it. We'll fill in some of these answers together. Okay, first question here. List 3 types of granulocytes. And as you might remember, the granulocytes are the cells that has visible granules when you stain them, when you look at it under the microscope. And we talked about 3 examples of the granulocytes. Right? So the first one is neutrophil. These are the most common phagocytes, and they go around and eat things that are not supposed to be there in your body. Next example is eosinophil. The eosinophils, they secrete chemicals from their granules and they attack things that are too big to be eaten up, to be phagocytized. So things like tapeworms, right? We would need to have some eosinophils to deal with them, okay? Parasitic infections in general. The last one that we talked about is something called basophil. The basophil plays a role in inflammatory response. Okay, inflammation. So those are the 3 types of granulocytes. We also talked about agranulocytes. These are cells that do not have visible granules. They have granules, but when you stain them, they just don't take up the dye as much, so you can't really see them too well. And we have 3 examples. Can someone give me 1 example of agranulocytes, please?
T cells.
Yeah, we have the, very good. We have the T cells, and I heard some of the B cells as well. So the T cells, they help coordinate the immune response. The B cells are the ones that will make antibodies. And then we have one last one here.
Monocyte.
That's right, Ziam. That is the monocyte. Very good. Monocytes, they are not capable of fighting infections. They're not capable of fighting pathogens on their own, but when they do encounter pathogens and things that does not belong to the body, they will transform, they will differentiate into macrophage, and those are capable of eating up things that does not belong to the body. Okay, so there you have the 2 categories of granulocytes and agranulocytes. Here is a quick matching question. Let's just take it up. Responsible for inflammatory response. Okay, we just talked about it. That would be the job of basophil. Okay, basophil. Coordinate immune response, right? Telling things what to do, telling the cells to transform, to change, to make antibodies. That would be the job of T cells. T cells. Can differentiate into phagocytic macrophage. That would be monocyte. Okay. Differentiate means it changes and it specializes. Okay, it's a one-way transformation. Once they become the macrophage, there's no going back to monocyte. Okay, it's just one-way ticket, if you would. Attack parasites. That would be the job of eosinophil. Most abundant phagocyte, neutrophils. And finally, responsible for producing antibodies, that would be the job of B cell. Okay. So these are all the Y cells that we've learned. Where do these Y cells come from? Just, to see if you remember. Where
Bone marrow.
The bone marrow, that's right. So in the bone marrow, the red bone marrow, right? There is a process, the stem cells, right? The stem cells will become all these things, right? And the process by which this happens is called hematopoiesis. Okay, not just white cells, right? Red cells as well, and the platelets. Okay. Let's take a look at number 4. List the steps in coagulation pathway, starting with the release of thromboplastin from injured vessels. Okay, so the thromboplastin will allow the conversion of prothrombin, which is inactive, right, into thrombin. And that requires calcium. And other important protein, plasma protein, that's plays a role in coagulation, is the fibrinogen. Okay. Again, ogen means it is inactive. And in the presence of thrombin and some calcium, it will get converted into fibrin. And the fibrin is what forms the mishmash, the net, right, that will catch red cells and platelets, and eventually it will form the clot. All right, moving on. Complete the following chart related to ABO and RH blood group. Okay. Remember, blood group is basically referring to the type of antigens that you have on your red cells. Okay. Type of antigens on red blood cell. There are different blood groups, right? 40 different blood group at least. The ones that we are focusing on is the ABO blood group and the RH blood group. So tell me, how many alleles do we have for the ABO blood group? How many alleles exist in the ABO blood group?
3 alleles.
3 alleles, that's right, Shelley. And they are IA, IB, and little I. 3 alleles. Okay. How many possible genotypes can we make up with these alleles? Remember, even though there are 3 alleles that are in the population, because we are diploid, we only have 2 sets of chromosome. At most, we can have 2 of the alleles. They could be the same, they could be different. So how many possible genotypes exist for the ABO blood group?
4.
4. I think you are thinking about the blood type themselves, right? You're probably thinking about like A, B, and O, right?
Sorry.
But that's okay. That would be the phenotype. That's the expression of the gene, right? But in terms of the genetic makeup, how many possibilities are there?
I think 6 genotypes.
6 is correct. Okay, so we have the IA, IA small I, IB, IB little I, IB, and little I, right? 6 combinations that exist. And the manifestation of these 6 genotypes is going to be the ultimate blood type, right? So you can be type A blood if you're IA or IA small I, so on and so forth, right? All right, let's compare this with the RH blood group. RH stands for rhesus, right? It's named after the monkey that was first identified in. How many alleles exist for the RH blood group? Let's think about that.
2 alleles.
How many, sorry?
2.
2, yeah. And they would be, we just call them positive allele and negative allele. Okay. The positive allele allows you to make the RH protein, and negative means you cannot make it. In terms of genotypes, right? What combinations can we generate? We can have positive, we can have positive, negative, and we can have negative, negative. Positive, positive, as well as positive, negative will give you Rh positive blood type, and negative, negative gives you Rh negative blood type. All right. In the past, students get confused because they start mixing biology with math, right? They're like, oh, negative, negative gives you positive in math, right? But remember, it's not related to that. Okay? Positive just means you are able to make the protein, and negative means you cannot. So if you inherited 2 alleles that does not allow you to make the protein, then you don't have it. When you don't have it, you are called Rh negative. Does anybody have any question before we move on? Great. Now, I asked you to fill in this chart, you know, on your own. I'm not going to take up the whole thing, but I will do maybe like a few just to, you know, help refresh your memory. When it comes to blood donation, the rules for ABO blood group is like this. Right, this shows you who you can give red cells to. And for the Rh negative people can give to positive, not the other way around. Okay, I'll tell you why later on. You probably kind of know why, but like I need to explain more because the story is more complicated. Okay. So like I said, right, have those nearby when you're doing your test probably will help. Okay. There we go. So if you're A positive, who can you receive red cells from? So we look at the ABO chart and we see everything that points to A, right? So only O points to A. So you can receive from O, and you can also receive from other A's, right? But you must also include the Rh blood group. If the person is positive, you can see here, they could receive from other positives, but they can also receive from other negatives as well. So A positive can receive from all these people. Who can you donate blood to if you are A positive? Well, A can donate to other A, and they can donate to people who are AB as well, right? You just follow the arrow, it will tell you who you can give it to. If you're positive, you can only give to other positives and not negative. Does that make sense? I'm going to pause for a second and just see if any of you have any questions. Okay, great. Let's do another one. Let's say we have AB positive. Okay, AB positive, who can you receive from? Well, AB can receive from all the blood type, right? You can see all these arrows pointing to it. So we have A, we have B, we have AB, and we have O, right? So it's positive, but you can also receive from the negative people. Okay, so AB positive, right? You kind of hit the jackpot. You can receive transfusion from all these people. And who can you give it to? You can give it to other AB positive. That's it. You can't give to AB negative because positive blood cannot be transfused into negative people, right? So AB positive can receive from all the blood groups within the ABO and the Rh, but they can only donate to AB positive. What about type O positive, right? Type O positive, who can you receive from? If you're O positive, then you can receive from O positive or O negative. And you will be able to give it to A positive, B positive, AB positive, as well as O positive. Okay. So you see, when people say type O is universal donor, that's not entirely correct, is it? Right? Because it might be true that type O is universal within the ABO blood group, but it's certainly not universal when you consider all the other blood groups, right? If you consider just RH alone, in addition to the ABO, you will see O positive cannot be given to, for example, an A negative person, right? So it's not really universal. It's only universal within the ABO blood group. Okay, so it's kind of like a misnomer. It's a wrong name. Are we okay? Let's just do one negative, okay? Let's just do B negative. If you're B negative, you can receive blood from O negative and B negative, right? You cannot receive from positive people. But now you can donate to positive people, right? You can donate to B positive, you can donate to B negative, you can donate to AB positive, as well as AB negative. All right. So do complete this chart and just try to figure out the logic behind it. You know, after you've done all of these, it probably will give you a clearer picture of what is going on. Okay, we did this already. Let's do one of these Punnett square question. Josh's father has type O blood. So let's draw Josh here. We have Josh and his father. His father has type O blood, which means the father must be little I, right? We don't know anything about his mother, and that's okay. Josh is going to marry Kat. And Kat has AB blood, which means she is IA-IB. And Josh has type A blood. So he's either IA-IA or IA small I, right? So I'm going to put IA. Because his father is little I, then it's guaranteed that the father will be passing on the little I to Josh. That's how I know he is heterozygous and not homozygous, right? Because like this little I get passed on to Josh like that. Okay, so now they're gonna have some kids, and we want to figure out what are the possible outcome. So Kat is IA-IB. I'm gonna put her gametes here. Half of it will have the IA allele, the other half will have the IB allele. As for Josh, he will produce gametes, sperms, that carry either IA or small I. And if you go ahead and combine the Punnett square, you will get this result. So there is a 50% chance that their kid is going to have type A blood. 25% chance for getting type AB, and 25% chance of getting type B blood. Okay? That's it. There's a Punnett square, a pedigree here. Has anybody tried this and want to share their answer? Just curious. If you haven't done it, that's okay. We'll take it up. But
Yeah, I did this.
yeah, do you have an answer?
Yeah, I got two possible blood type, which is IA-IB and IA small I.
IA. Okay, thank you. Anyone else have any other answers? Or do you agree? Do you have different ones that you want to share? I don't remember what it is. Like, I have to do it with you right now, but just wanted to throw it out there in case anyone else want to share. Okay, that's fine. We'll figure it out. So it's not going to get harder than this, right? Like on the test. If you're able to solve this question, you are in a good place. So we will start with, blood type that we are certain about the genetic makeup. There are only 2 blood types that we can know for sure, which is A, B, and O. So in this case, we got a type O lady here, and she's going to be little I. Everyone else, we only know half of it. So IA something, IB something, IB something here, and IA something here. All right. And we gotta, use a little bit of logic to figure out the rest of the answer. Now this lady has little I. So one of the little I must have came from the father, and the other little I must have came from the mother, right? Because you get half your genes from each parent, right? So that's pretty straightforward. So let's do more detective work here. We have an IA over here, an IA allele. Where did the IA came from? Did it came from the father or the mother? Well, the father is type B. It's impossible for him to have an A allele, right? Based on that reasoning, we know this IA must have came from the mother. Okay. There we go. But what about the other half of the genetic material? Well, where did question mark got her IA? If you look back to her parents, it has to be the father, right? Now, based on the current result, current family tree, we do not have enough information to know whether the question mark inherited IB from the mother or little I from the mother, right? We just don't know. They have to have more kids for us to be certain. But for now, based on this information, question mark can actually be IAIB or IA little I, right? So Shelley's correct. That's the answer. Do you guys understand why it could be both? Okay, so let's say they have another kid, right? Let's say they have another kid. Let's say this time they have a boy. And the boy is AB. Will we be able to tell now whether question mark, let's give her a name. Let's call her Jane or something. Do we know if Jane is going to be IAIB or IA little I now, if they have another kid that's AB? Let's have a vote. If you think you can tell now, well, tell me what the blood type. If you think she's IAIB, choose A. If you think she's IA small I, choose B. If you think is still can't tell, cannot tell, not enough information, please choose C. Okay, I'm saying they have another kid now. Okay, they have a little boy, and the little boy is AB. Will I be able to tell now? Okay, thank you to those who have participated. Yeah, it's kind of all over the place, right? We have people choosing each of these choices. The correct answer is we still can't tell. Okay, and this is the reason. If the boy is IAIB, right? The A could have came from Jane, and the B could have came from Dad, right? Okay. I mean, using the information that we have right now, not knowing what the other half is, right? We are still able to create a child that's IAIB. And that's why we cannot definitively say whether she's IAIB or IA small I. I understand why some of you might think she's IAIB, because you're thinking, well, you know, she probably needs to have a B in order for us to in order for her to have a boy that's AB, right? But not necessarily, right? The B could have came from the father, right? And not necessarily her, right? So that's why we still don't know. Okay, then what kind of kid do they need to have in order for us to be able to definitively say whether it is one or the other, right? What will there be a child with a certain blood type that will help us decide whether she's AB or AI? Anybody want to try?
Oh,
AB small I.
Okay, so I hear a couple answers here. We wouldn't know if the person is IB little I, right? If the person is type B blood, then it could be IB or IB small I. So having another child that's type B blood will not help us determine definitively whether she's AB or A. Okay, and then I see people typing O, and I heard someone said O as well, and that would be correct. If they have another child that's type O blood, then it means the child is little I, right? Then that guarantees she's going to be IA small I. Does it make any sense? I hope it does.
I have a question.
Yes, please.
So the type B father, wouldn't it just be BI? Because if it was BB, then they couldn't have a type A.
Yeah, that is also an excellent point that you made right there. Leah is saying, you know, let's forget all these extra things that we've added. Okay, so she's saying if this person is type A, then doesn't that guarantee the father has to be IB little I, right? Because if he's IB, then it's impossible for this person to be type A blood, because he would only be able to pass on IB, right? So yeah, you're absolutely correct. So we do know for certainty that this is heterozygous. Okay. All right, I have another question in the chat. Let me see, what is it? If the baby is O type, their parents have to be IA little I, IB little I. That's correct. Okay. And then the next question is, if the baby is AB, there are more options. If we didn't see all the answers, we will choose not sure. Yeah, okay. So like on the test, if I throw you something like this, there will always be a choice that says like not enough information. Okay. Then you would definitely choose that as your answer. But we did some last time, right? Something like this, right? So like I would give you things like, for sure they're, you know, AB, for sure they're IB little I, either this or that. And then, you know, if it's none of these, then you say need more information. Okay. But it's not going to be more complicated than the ones we've done. Okay. Just practice the pedigrees that I've gave you. There will be no surprises. Okay. If you know how to do all these pedigrees that we did together, you're going to be okay. All right, moving on. Actually, give me 1 second. I just need to fix something here. Okay. This is a baby matching question. You have 4 parents here, and you know, somehow the hospital mixed up all their babies. We got to figure out which baby belongs to which parents. Okay, that's what we have to find out. Now, when you do something like this, you know, many people would start drawing pedigrees and stuff like that, and then it would take forever to do this question. Okay, you don't have a lot of time on the test, right? So what you want to do is you want to remember 4 things, that would help you in this situation. Okay, so I'm gonna write down 4 things to remember for matching question. For matching babies and parents questions. And there are 4 things you should remember to help you solve these questions easily without doing Punnett squares. Okay, number 1. A person with AB blood cannot have kids with what kind of blood? Okay, I'm saying if you have AB blood, it doesn't matter who you marry. Okay, what kind of kids will you never be able to have? Okay, Jamon said O in the chat, and that's correct. And you will never cannot have kids with O blood. That's because if you need to have a child with O blood, you must be able to contribute. You must be able to pass on a little I. You don't have a little I when you are type AB blood, right? So that's why it doesn't matter who you marry. Right? You're not going to have a biological child with type O blood. Okay. Can someone else give me another statement that's true? Along something along this line? And another generic statement that would be true. I'll start you off. Yep, go ahead.
Okay, two parents have if both two parents have negative blood, cannot have a baby with positive blood.
Yes, that's good. That's actually the 4th one that I want to say. Okay, but we'll write that down first. Okay, so 2 people, 2 Rh negative people can only have Rh negative babies. If the father is Rh negative, the mother is Rh negative, then the whole family, it's going to be Rh negative. All right. Another one that's along this line, a person with what kind of blood cannot have kids with what kind of blood?
A person with O blood cannot have kids with AB blood.
That's right. The converse is true, right? The opposite is true. If a person with type O blood, they are little I, and in order for them to have a child that has a AB blood, you have to either contribute an IA or you have to contribute an IB, right? Do you understand? Okay. Now I'm just gonna say it out loud for you to think about this. You know, at first glance, the following statement sounds true, but actually is not true. Okay. Some people might think a person with type A blood cannot have a kid with type B blood. Right, let's think about that. If you have type A blood, then it's not possible for you to have a kid that has type B blood, right? That actually is not true, right? Because if you're type A, you could be IAIA or IA small I. So if you're heterozygous, then yes, it's possible for you to have a type B child if you marry someone who is type B blood or type AB blood, right? So that's not universally true. That's why we don't include it in these statements, right? Okay, these statements are always true regardless of who you marry, and you know, there's no confusion about whether you're homozygous or heterozygous, because AB blood is just one type of genotype, right? Same thing with type O. One last one here. Anyone else want to try? Help me out. It's similar to number 4, the one that I'm looking for. 2 something people can only have something babies. What do you think, huh?
2 Rh positive people can only have Rh positive babies.
Okay, thank you for trying and saying that. Again, that's very common for many people to think. If the parents are Rh positive, then the whole family is going to be Rh positive, but not necessarily. Okay, because the parents, if they're Rh positive, they could be heterozygous positive, right? And in this scenario, there's a 25% chance they could have an Rh negative child, right? That statement would only be true if they are both pure positive. Okay, then the whole family will be positive, right? But just by saying someone is Rh positive, there's no way for us to tell immediately whether they are homozygous or heterozygous. So we cannot generalize like that. But thank you for pointing that out. I'm sure many people felt the same. Yeah,
Can I go? Oh,
please go ahead.
AB people can only have A or B babies. You can't have O. Or so if two people have A and B, because it's co-dominant, they can't have O.
But okay, yeah, what you said is true, but that is already included in the first statement, right? Right. Think about it. Right.
Wait, is the A person like the first parent with A, or you're referring to A, B, and A as a parent?
Say that again. Sorry, I didn't quite catch the question.
No, I think I wrote my note wrong.
Oh, okay.
So when you say a person, do you mean like the person is an A, or just?
Oh no, like a person, like an individual. Yeah, I see why there is a confusion.
Oh, sorry.
Sorry. Okay, I see. Emma said 2 O people can only have O babies. That's right. That's the one I'm looking for. Okay, if the parents are type O, right, that means they're little I, right, and then the whole family will be little I, right, because that's the only thing that could pass on. Okay, I want to make sure that everybody understand these 4 statements, because I will give you matching questions on the test. Okay, and if you do it with the Punnett square, you will definitely run out of time. So I just want to make sure that there's no confusion about these statements and why they're true. And I think this is an exhaustive list. Okay, I don't want you to make up your own rules. I mean, even though your rules might be correct, but it probably will not apply universally. Okay, these rules apply universally in all situations, regardless of who you marry, and regardless, you know, we don't even have to think about homozygous or heterozygous, because there's no such thing for people with AB blood or with O blood. Okay, let's put these rules to the test. And you know, we'll do one question and see if it makes sense, and we'll come back to it if there are any questions. All right, so let's take a look at this you know, baby matching question. How do we do this? Well, I always start with parents that have, you know, at least one of them is O, or at least one of them is AB, and we use logic and process of elimination to try to solve the question. So let's say we start with couple number 2 here, right? Okay, listen to me carefully. The mother is O and the father is O. Okay, that means I can apply rule number 3. People who are O, 2 O people will only be able to have O babies. So I know it's not baby H, I know it's not baby J. It has to be baby I or baby L, right? Then we look at the RH group. It's negative and negative. 2 people who are negative will only be able to have negative babies, right? For that reason, guarantee if these are all the babies up for grabs, L belongs to them. Do you understand how I use the rules to solve this question? Right? You don't have to draw the alleles. You don't have to do Punnett square, right? Okay, so baby L is gone. Baby L went home. Good for it. Next one. A, B, and O. All right. So if the mother is AB, what kind of babies can she not have? Think about that. What does the rule say? AB people cannot have O babies, right? So from the list here, we would take out baby I. That's not hers for sure. The father is O, and O people, like type O blood people, cannot have AB kids, right? So that's not their baby. And so baby H belongs to them. All right, let's take a look at number 4. The mother is O. We only have 2 babies left, right? Type O blood individuals cannot have AB baby. So that's not theirs. And that means it's baby I. So after baby I is taken, there's only one baby left. That must be theirs. Are you with me, guys? Any questions that you want to ask? You know, students often tell me, oh, it looks so easy when you do it, right? And then when you try it, you always run into something. It's true. Like I try to watch YouTube to like fix the leaking faucet or something, and then it makes it look so easy, and it's just a 3-minute video. And then when I do it, it takes like over an hour to do it. But you know, as cliche as it sounds, practice makes perfect. So why don't you try this one on your own? Okay, I'm gonna give you like maybe 2-3 minutes to try this out, and then we will take it up. All right. Okay, does anybody want to share their answer? You can just tell me like the letters. You can type in the chat box, okay? Like you can just type like JLHI, but like with these letters, right? If you have the answer and you would like to share, please type it out. Just type out the letters, okay? Like is it you know FGHE, EGFH, whatever, right? Like just type it out. Okay, I have answer from Zia. Anyone else want to share? Leah also got the same thing, it looks like. Okay, Clara has something that's different. Okay. Okay, great. Let's try to take this one up. Okay, good. A lot of answers coming in. Thank you. So same thing. Okay, let's start with people with type AB or type O blood in the parents. And the first one here we have AB. So AB cannot have O babies. That's not theirs. Oh, sorry. AB cannot have O babies, so that's not theirs. Type O cannot have AB babies, so that's not theirs. They are both negative, so you cannot have positive baby. So that's H, right? The first couple, baby H is who they're gonna take home. Okay, baby H is gone. Next one, O cannot have A, B, so we cross that out. And then we have two choices left, O positive or A positive. So at this moment, I don't have enough information to rule out one over the other, right? I just both are possible at this moment. So I'm gonna put down F or G. We're gonna come back to it later and see if someone else will claim one of these babies, okay? O negative and B positive. If you're type O, you cannot have a B baby. In this case, the mother doesn't have an A and the father doesn't have an A, right? So it's impossible to have a child, right? So baby G is not theirs. And so F is going to be their kids. And since F is taken, then you know the previous couple, G would have to be their baby. And once these three are settled, then E is going to be the fourth couple. So the correct answer is H, G, F, and E. Okay, so I hope that makes sense. Again, try to practice some of these things. And you know, you may want to even try to come up with your own questions. You know, actually, you know what? If you come up with a nice matching questions, you can email it to me, okay? And then I will add it to the practice for people to do. You know, in making it up actually will help you understand the concept better, because these things are not easy to make up, right? Like you got to make sure there's no redundancy, and then they actually all cancel each other. So if you're able to create one and it all works out, it really helps you understand it a little bit better. And I could use more questions, you know, for people to practice anyways. So if you make one up, or if you want me to check it over, please do email me. Okay, we cannot do any of these things yet. We will take a new class notes. All right, so this next thing is also quite intense, right? A lot of explanation, a lot of concepts to go through. So please Please pay extra attention. Okay, so this concept that we're gonna be talking about now is RH mismatch. Okay. So there is something that you should know, right? Just think about what we learned last class first, right? You don't have to write this down yet, okay? I'm just trying to recall some information to bring you up to speed. So let's say you are a person with type B blood, right? Just remind me, what kind of antibodies are you gonna make in your system? What kind of antibodies do you have regarding the blood groups? Anti what?
Anti-A.
That's right, anti-A, right? Everybody agrees with this, right? Okay. You make antibodies for things that you do not have. Okay. Now, for all intents and purposes, right? For our course, you can assume that if you are type B blood person, you are pretty much born with the anti-A antibody. Okay, it comes with you when you come into this world. All right, but that's not really how antibody works in general. Okay, this is just like an exception, right? That you're born with the anti-A antibodies. If you think about it, like my favorite example, right? Chicken pox. Okay, how do you make anti-chicken pox antibody? What condition needs to occur first before you can make anti-chickenpox antibodies? Are you born with anti-chickenpox antibodies?
You have to contract the virus.
Exactly, right? You got to get chickenpox first, and then you make the anti-chickenpox antibodies, right? You either get the virus through natural means, or as we'll learn next class, you can get vaccinated, and then you will get anti-chickenpox antibodies, right? My point is, the antibodies that our body makes is not something that we're born with. Okay, for the ABO blood group, you know, that's just an exception, and we're not going to go into the details of why and so on and so forth. Okay, how does all this related to what we are talking about? So I'm going to write here, unlike you know, anti-A, anti-B antibodies, an Rh negative individual will only make anti-Rh antibodies after they are exposed to Rh positive blood. Okay, so that's kind of like the chickenpox deal, right? You can't make chickenpox antibodies until you've exposed yourself to chickenpox. In the same way, if you are an Rh negative individual, you will not make anti-Rh antibodies until after you are exposed to Rh positive blood. Okay, so that's the first thing you must understand before we move further. Okay? It's not like the ABO system where you have those antibodies when you're born. Okay, for Rh is different. Okay, you must be exposed to Rh positive blood. Okay. So we're gonna have two examples of Rh positive blood exposure. Give me some examples, guys. Think about it. Why would an Rh negative person be exposed to Rh positive blood? What are some ways of this happening? Realistic ways that happens in the real world.
The negative person received the blood donation from positive person.
That's right. Okay, so this is a blood transfusion. Rh positive blood is transfused. It's transfused into an Rh negative person, right? Now, I know what you're thinking. You're like, wait, hold on a minute. Didn't we say positive blood cannot be given to a negative person? That is true, but hang on tight, and we're gonna get back to that in a second. Okay, but positive blood is sometimes given to a negative person, either by mistake or sometimes on purpose. Okay, because they don't have other choice. And I'll explain more. And any other possibility? Do we have any mothers here? Encountering any Rh issues during pregnancy? You might probably know about the second reason. No? All right. The second possibility is an Rh negative mother carrying an Rh positive baby. And then the baby's blood got into the mother. That could happen, and I'll explain more later. All right, so these are two examples of how an Rh negative person can be exposed to positive blood. Okay. Now I'm going to explain a few more things, and it will all make sense in the end. Okay, I promise. So when an Rh negative individual is exposed to Rh positive blood, either through wrong transfusion or the mother-child situation, the person is, we have a word for this, sensitized. Okay, sensitization. Okay, so that's like when you're exposing the negative person to positive blood. Now, what does it mean? Once sensitized, the person's immune system starts to make anti-Rh antibodies. Okay, so that's what sensitization means. Now, listen and see if this following explanation makes sense to you. Consider an Rh negative person that has not been sensitized before. Okay, so this person has never been exposed to positive blood before. Is it okay to give them positive blood just for once? Okay. Is it okay to give them Rh positive blood one time?
And how long would it take for them to be sensitized?
Oh, that's a good question. I forgot to mention that. Yeah. So you don't make the antibodies like right away, right? Think about it, right? When you get the cold or when you get the flu or COVID, your body doesn't make the antibodies immediately to fight it off, right? It takes about a week or so before you start feeling better, right? So that's usually how long it takes to make the antibodies. Does that answer your question?
Yeah.
Yeah. So right, like back to our scenario, an Rh negative person who has not been previously sensitized, is it okay to give them Rh positive blood like just once? It's okay, right? It's okay, right? Because the person doesn't have the antibodies yet, and we can give them the positive blood, and the blood will save them if they're losing blood, if they're bleeding out, right? Okay. So that's why I said sometimes, you know, people do it on purpose, even knowing that the person is negative, right? And if there is medical record that shows the person was not previously sensitized, right? And they don't have negative blood around, it's okay to give them positive blood like once. Okay,
So what would happen if the person gets the positive blood and then they're still not sensitized even after receiving the blood?
okay, that's a great question, right? Now, once you receive the positive blood, you will become sensitized, right? Like your body will react to it and make antibodies, but just not like right away, right? It takes like about a whole week before, you know, you actually get all the antibodies being made. So what that means is all the people who are Rh negative, they have one chance of receiving positive blood, okay? But then if they need positive blood, if they need blood again, you know, after a few weeks, then you cannot give them positive blood again because you already sensitize it through the first transfusion, right? So all the negative people will only get one chance, one free chance of getting the positive blood transfusion. Once sensitized, you will not be able to receive positive blood again because you will have the antibody in your system now, right? And then those antibodies will attack all the positive cells that are coming in, right, resulting in hemolysis. Does that make sense, guys? This concept of being sensitized and, you know, receiving positive blood and so on and so forth.
So basically, you can only be sensitized once.
If you are sensitized, you are forever sensitized, right? Right? You're not going to get sensitized again because once you've exposed to the positive blood, you will forever have the antibody in your system, right? Just like you only get chickenpox once, right? You're not going to get it again because you have the antibody in your system already, right? Does that answer your question?
Yeah, so does that mean in the future then you can receive positive blood?
You cannot. You cannot,
Okay.
right? Right? Because you already have the antibodies. If you receive positive blood again, those antibodies will reject the positive blood, right?
Okay.
Right? It was okay to receive it the first time because you don't have the antibodies yet, right? Okay, hopefully that makes sense. Now we're going to talk about the mother and the child situation, and that could really, you know, cause a little bit of confusion. So draw along with me, ask questions, you know, along the way. You guys are awfully quiet. A lot of you, I haven't even hear your voice yet. So don't be afraid to ask questions, okay? Sorry. Okay, so someone asked, is it what you, okay, so they're asking about vaccines, and, you know, we're going to talk about vaccines a lot next class. So we'll save that for later, okay? But similar concepts here. Okay, what was I saying? Okay, so let's consider this scenario, okay? Consider a couple. Okay, so we have the mother, and, you know, just for simplicity's sake, we're going to say the mother is Rh negative, and then the father is, you know, homozygous positive. Okay, this could happen to heterozygous father as well, like positive/negative. But we're just going to keep it simple, positive. Okay. So let's say they have a kid. What's the Rh status of their child? What kind of kids are they gonna have? Work it out for me, please.
Positive?
And is it guarantee, or like, is it 50%, 25%? What are we talking about here?
Oh no, it's guaranteed because it doesn't matter.
Yeah, exactly. The father is guaranteed to pass on a positive, which will override the mother's negative, right? So 100% guarantee the kid is going to be Rh positive. Okay. All right. So let's consider the first pregnancy. Okay, and this mother has not received a positive transfusion or anything like that. She has not been previously sensitized. Okay, so you know, this is the mother's bloodstream. Please draw along. Okay, mother's blood, and you know, we're gonna have the baby here. Okay, the fetus, right? The baby the baby's blood. And what connects the mother and the baby?
The umbilical cord.
Yes. And then the umbilical cord is attached to what in the other end?
Placenta.
Placenta. That's right. Placenta. Can't spell this morning. Placenta. Yeah. Okay. Wait, that doesn't look right. Sorry. Placenta. There we go. There we go. Placenta. Okay, so placenta is the thing that connects the mother and the baby, or supplies the nutrients and all that stuff. For those of you who don't know, when after the baby comes out, the placenta also comes out afterwards. Anyways, so the placenta does a pretty good job separating the mother's blood from the baby's blood. Okay. In our scenario here, the mother is Rh negative and the baby, thanks to the dad, is Rh positive. So we're gonna draw some red cells. Okay, so for the mother, we're just gonna have happy faces without the eyes. Those are our red cells. Okay, so these are Rh negative RBC. And for the babies, we will have blood cells, but because they are Rh positive, we're gonna put Rh on them. Okay. So like I said, the placenta does a pretty good job separating the mother's blood cells and the baby's blood cells. There is no mixing, right? The nutrients can cross the placenta, the oxygen can cross the placenta, but we keep the red cells separate. But, you know, things can get complicated during pregnancy. Okay, maybe there are some accident that could cause the placenta to be a little bit damaged. Okay, maybe there is some tearing going on, and the baby's blood might go into the mother's bloodstream. Okay, during labor, when the mother is actually giving birth, right, that could cause some damage to the placenta as well, and some of the baby's blood might go into the mother's bloodstream as well. Okay, so during, you know, my point is this: some complication occurs, and then the baby's blood is going to go into the mother's bloodstream. Okay, the complication causes the placenta to damage, right? So now what happens? Well, what happens next is the baby's blood cell is in the mother's system now. And then what's going to happen to the mother? What's the word that we're going to use to describe the mother's situation?
Sensitized.
That's right. The mother is now sensitized, right? Okay, well, what does it mean? It means the mother will now make anti-Rh antibodies. Okay, now let's say this happens during labor. Is that going to be a problem for the baby? That
No.
the mother, why not?
Well, if it happens during birth, then she's sensitized after the baby's out, so she's not going to be exposed anymore to that.
Yeah, and just to clarify what you're saying, I think you're on the right track. By the time the mother makes the antibodies, right, the baby is already delivered, right? So those antibodies is not gonna harm the baby. You understand? Right? Because the baby is already out, right? Okay, it takes like about a week or so, maybe longer to make those antibodies. And by that time, the baby is already out. Okay, so does everybody understand the scenario here? Before I move on, the story is not over. All right. So let's say the baby has been delivered and everything is okay. All right. I'm just gonna erase some of the other stuff here. Let's say we go to the second pregnancy. All right, you probably have to redraw this later on. Okay, but just try to understand what I'm saying now, and then you can revisit the recordings later and add what you need. Okay, so second pregnancy, right? Let's say the mother has another child with the same father, which guarantees the kid is going to be positive again, right? So here we are, the exact same drawing, but there is something different, right? There is going to be something extra that wasn't there during the first pregnancy. And that would be the antibodies, right? Because the mother was sensitized during the first pregnancy, she will have anti-Rh antibodies in her system now. So during the second pregnancy, what happens is some of these anti-Rh antibodies will cross the placenta. And then it will attack the baby's red blood cells. Okay? And that is bad news for the baby, right? Because now the baby will experience massive hemolysis. Okay, a lot of the red cells will die in the baby. Right? So when we break down red cells, the hemoglobin comes out and that get metabolized, and then there will be a lot of bilirubin. Okay. And if this happens continuously up to the point of birth, then the baby is going to be born with jaundice, right? Because there's no way to deal with all these extra bilirubin, makes the baby's skin tone very yellow, and then you got the jaundice. And there is usually enlarged liver and spleen, right? You remember I told you the spleen and the liver is responsible for destroying old or worn out red blood cells, right? So all these red cells that are dying, right, it causes the liver and the spleen to work overtime to deal with them. And so they have enlarged liver and spleen. And when all these red cells are dying, then your blood concentration, right? Remember the osmolarity, it's a combination of all these things that are within the cell, within the blood, right? And so if you're losing a lot of these red cells, then it causes you to not be able to retain the fluid within the blood vessels, and it causes swelling all over the place. Okay, so these symptoms could be very severe if the reason is because of Rh mismatch. Okay, and this is not restricted to the Rh blood group. It can happen to any of the blood group if there is a mismatch, but the severity does vary depending on which blood group you're talking about. So collectively, we call these hemolytic disease of newborn. Okay, HDN, hemolytic disease of newborn. Any questions before I move on?
Why the baby?
Sorry, go ahead.
Go ahead, Shelly, and then Lashana. Yeah.
Why the baby have more bilirubin?
Because, okay, so like all the red cells are dying, right? When red cells break apart, the hemoglobin comes out, right? Remember in the lecture, we said the hemoglobin gets broken down, right? You recover the iron, and then the rest becomes the bilirubin, right? That's like normally what happens, right? If it's not excess amount of bilirubin, we just get rid of it from from our urine, from our feces, and so on and so forth. But when there's so much red cells dying, then your body cannot get rid of it fast enough, and so the bilirubin builds up. Okay.
Because of the red blood cells, a lot
dying.
of them.
Exactly. Yeah.
I understand. Thank you.
Yeah. Sashana, go ahead.
Oh, I was asking, so what happened when the baby's born with jaundice, but not with the enlarged liver and spleen and stuff?
Okay,
What's
so
that
with
because of?
just the jaundice, it could be a mild case, and I'm gonna get to that in a second. Okay, just hold that thought. We'll come back to it because I have a slide coming up for that. Anyone else have any other question about this? Okay, usually people would ask, is there anything you can do about this, right? That's a logical thing to think about, right? Okay, like if you don't have kids right now, and you plan to have kids, and you are a negative person, and maybe your spouse is positive, right? This is a real concern for you, right? Like this could happen to anyone who's negative, and then the spouse is positive, right? So is there a way to prevent this? And the answer is yes. Let me see if I have it in the PowerPoint or if I have to draw it out. Okay, so this is professional drawing, right? Of what I was trying to show you earlier. The first pregnancy, you can see there's no antibodies in the mother, but then giving birth, because there was hemorrhaging, bleeding, right, that damaged the placenta, then the baby's positive cells with all the spikes goes into the mother, and then the mother will now have antibodies. In the second pregnancy, the antibody crosses the placenta and it causes the baby cells to be destroyed, hemolysis. So that's basically the problem with hemolytic disease of the newborn. Okay, there's also like a fancy name for it, right? Erythroblastosis fetalis, but we're just gonna call it HDN for short. Like I said, the symptoms severity varies, and it might include anemia, jaundice, enlarged liver, spleen, severe edema, so on and so forth. And sometimes it could be lethal, right, for the baby, if it's severe enough. Okay, so how do you prevent something like this from happening? Okay, so there is something called RhoGAM. RhoGAM. Okay, a RhoGAM is artificial anti-Rh antibody. Okay, artificial means it is man-made in the lab, and they are anti-Rh antibodies. The purpose of RhoGAM is to prevent sensitization. Okay, the key word here is prevent. Okay, not reverse, not cure. All right, if you already got sensitized, giving you RhoGAM will not reverse the process. Okay, this just prevents it from happening. Right. So how does it do it? Okay, listen. Let me go back to here. Right, so
Sorry, Professor, quick question.
Sure, please. Yeah, go ahead.
So if the sensitizing happens during pregnancy, how would they know?
How would they know? They would check on the baby, right? It's possible to it probably will show up on some blood tests or some scans that something is happening. They can also test the mother's blood, right? To see if there is any Rh antibody. If they suspect this is something that's happening, they could easily test the mother's blood for Rh antibodies, right? Okay.
No, but quick question, would something be happening to the mother for them to have this suspicion to say, okay, let's check for this? Or this is just a routine check that they check?
I don't think it's a
during
routine check that they
pregnancy?
check for. And after I explain this, it might make more sense, okay? And if it
Okay.
doesn't explain it, you can ask the question again. So let's say this is first pregnancy again. Okay, I know I'm changing the note a lot. You probably have to organize it afterwards. Okay, so let's go back to the first pregnancy. Now, a lot of times, especially like in Canada, for example, it's very common for mothers to go in and do some blood tests. Sometimes if it's planned pregnancy, they would ask you to do some blood work, right? Before you conceive, just to see if you potentially will run into this problem, right? And so, if the mother is negative and the blood test of the father shows that they are positive, then the doctor would recommend getting the RhoGAM shot. Okay, so first pregnancy, same situation as before, but now we're gonna give the mother the RhoGAM. Okay, so I'm gonna draw the RhoGAM here. This is the anti-Rh antibodies from the RhoGAM. RhoGAM. Okay, so what does it do? Well, let's say the placenta is damaged, and then some of the baby's blood is once again going to go into the mother's bloodstream, right? So I'm going to draw a baby blood cell here, baby blood cell here. But now, because the mother has the RhoGAM antibodies, the RhoGAM antibodies will actually destroy all these positive cells that are coming in from the baby. It will destroy them before the mother's immune system is triggered. Okay, so by mopping up all the positive cells that are coming in, you prevent sensitization from happening, right? Because you don't give a chance for the immune system to be triggered, right? So the mother will not be sensitized. So the second pregnancy comes along, right? It's not going to affect the baby because you were never sensitized. You wouldn't have those antibodies in your system. Does that make sense? Usually there is a question here about something.
So it's like an artificial decoy.
Yeah, that's a perfect way of putting it, right? Like you have the antibodies there to act as decoy to make sure that none of the Rh positive cells coming in will trigger the alarm, right? You mop them all up before the mother is sensitized. Okay. All right, now usually people would ask this, but why wouldn't the RhoGAM cross the placenta and attack the baby's blood? Isn't that what the problem was, right? These anti-Rh antibodies crossing the placenta and causing hemolytic disease of the newborn? Like, that's a valid question, right? Think about what I just said, right? Wouldn't these antibodies cross the placenta, right? So the answer is of course not, okay? Otherwise it wouldn't be a medication. This is artificial antibodies, right? Without going into too much details, you should know that there are different kinds of antibodies. Some are really big, some are really small. So they purposely make these RhoGAM, these artificial antibodies really, really, really big. So big that they are not going to be able to cross the placenta. Okay? They will remain within the mother's blood and not harm the baby at all. Okay? Another thing is these antibodies are not permanent. Okay, so you receive the RhoGAM shot, eventually the antibodies will break down. So in future pregnancies, you will also need to require another shot of RhoGAM, right, just to prevent sensitization. If sensitization already has happened, this is not going to do anything for you, okay, because you already made the antibodies, and those antibodies will still cross the placenta and affect the baby. Okay. So, to answer your question, Tashana, is this something that they regularly screen for? No, not really, because they would have already known the mother is negative and the father is positive, and they would have suggested to give them the RhoGAM in the first place, right? Okay, to prevent this from happening at all. So when do you give them the RhoGAM? You would give it to them at 28 weeks of pregnancy, and then you will also give it to them after childbirth, right? Just in case there was some hemorrhage going on during the delivery and there is some mixing of blood, you want to mop up all those positive cells coming into the mother. Okay. Now, it's also important to say that just because you're negative and your spouse is positive does not guarantee sensitization, right? The placenta might be perfectly intact during the whole duration of the pregnancy as well as during delivery, right? But there is a chance of sensitization going on. Okay. Now, like I said, the severity varies from depending on what kind of blood group mismatch we're talking about. It could actually happen for type O ABO as well. So these are actually my kids, my daughters, and they were both born jaundiced. The second one was very jaundiced compared to the first one, and it's because of ABO mismatch. I am AB positive, I told you. My wife is O positive, and the babies are B positive. Okay, so B positive for both of them, and my wife is O positive. So what kind of antibodies does my wife have? Well, if she's O, then she has anti-A and anti-B, right? So some of her anti-B actually was crossing the placenta and attacking the baby's red cells. But the severity is very mild. Okay, that's why there's only the jaundice that shows up, right? It's not as severe as the Rh mismatch. Okay, and so the way to fix this problem was to put them in this tanning booth, essentially. Okay, so using the UV light, it will help them break the bilirubin down, okay, and then get rid of the yellow hue, okay, so that their system doesn't have to work as hard. So my first one was only it only took like 2 sessions of these Billy light. It's called Billy light. It's basically UV light, right? Like I said. But then the second one, It was okay after 2 sessions, and then we went home, but then she keeps on still getting very yellow. And then we went back, and then she has to do it for another week. And the reason, part of the reason for that is, even though they're both positive, it was more severe for the second baby, because the second one didn't want to come out. And for those of you who don't know this, when the baby refuses to come out, you have to use a vacuum to kind of suck them out. And that's what happened for her. When you use the vacuum to pull them out, it creates a really big bruise on their head. And that bruising contributes to red cells damage and elevated her bilirubin even further. And so, it took longer for it to clear up. You might have heard of people saying, just leave your baby in the sunlight, and it will help with the jaundice. And that's true as well, right? Sunlight contains UV, and it's the UV that's breaking it down. Okay, they're both fine now, of course. Grew up completely normal. And as I said, the severity varies for some of these conditions. I have a question in the chat, and then I'm going to give you a break. Okay, what if the scenario is vice versa, where the mother is positive and the father is negative? That's not going to be a problem. Okay, positive people will never get sensitized. If the mother is positive, it doesn't matter if she's carrying positive baby or negative baby. She will never make the anti-RH antibodies, right? So this is only a problem if the mother is negative and she's carrying a positive child. Okay, that was a lot of information, and I've been talking for way too long. So let's take a quick break. We'll resume at 10 o'clock, okay? I'll give you a little bit longer to, gather your thoughts and get a break. And we'll do more afterwards, okay? See you in a bit. Right, let's move on to the next concept. This is something that we normally would do in the lab, like, if we were doing this, if we were having in-person lessons. But I'm just gonna go through the theory with you. So we have a blood sample here, and we read the thing across, okay? There are 3 wells here. The first well contains antibody A. Second well contains anti-B antibodies, and then the last one contains anti-D. Anti-D is anti-RH. It means the same thing, okay? Anti-RH. So what you're trying to determine in these blood tests is whether or not the antibodies are reacting with your blood sample. Okay, we want to see if the antibodies are reacting with the blood sample. So how do we know if there's a reaction or no reaction? If the antibody recognizes the red cells, there's going to be cross reaction, right? We talked about that last class, and that would lead to agglutination or clumping. So over here, you can see all the cells, or all the red cells are clumping together. That tells you there is a reaction. Okay, so let's interpret the result. The blood sample reacted with antibody A, which means the blood is going to have A antigen on it. Okay. It did not clump with antibody B. That means there is no reaction with the antibodies. And that means there is no B on your red cell. There is reaction with RH antibodies. That means there is RH on your red cells, and that makes it positive. Okay, so that means the blood type is A positive for the first example. Let's try another one. No clumping between antibody A and your sample. That means the sample does not have A on them. There is clumping with antibody B. So it's going to be type B. clumping with RH, that means it's positive. So second one is B positive. What about the next one? Why don't you give it a try and let me know what you think? What is the blood type of the third one?
O positive.
O positive. Okay, anyone else? Min said AB positive. Okay, so we have O positive, we have AB positive. Anyone else?
O positive.
O positive. Okay. All right. Let's see. So, did the antibody A reacted with your sample? It did, because I see the clumping, right? Okay, so that tells you that there is A in your sample. The antibody B reacted with your sample. That means it tells you there is B as well. And then it reacted with the RH, so it's AB positive. For any of you who's thinking this is O, I understand what you are thinking, and you shouldn't think like that, okay? You are thinking a type O person has antibody A and antibody B. I assume that's what you're thinking, right? And that's how you concluded it was type O. This has nothing to do with that. This is talking about you have a well, a plate that already has the antibody, and you are only adding the blood sample to see if it reacts with the antibody or not. Okay. When there is a reaction with the antibody, it tells you what kind of antigens you have on your red cell. So if there is a reaction with antibody A, then you must have an A on your red cell. If there is a reaction with the antibody B, then you must have a B on your red cell. And if there's a reaction with RH, then you must be positive. Any follow-up questions?
Sorry, what was the medical name for the clumping?
Oh, agglutination. I'll write for you. Okay, it's in one of the earlier slides. You can look back later. What
I
about
have
the
a
last? Sorry, go ahead, please. Yeah, sorry.
No, sorry, just a quick question about, so the blood sample, when it reacts to the antibodies, let's say the first one, A, right?
This one?
A
Yeah.
would have anti-B, so wouldn't it react with
Okay.
B?
Yes, that's again, right? Your blood sample is just going to be the red cells, right? Just the red cells. So in this well is going to be anti-A antibody. So when you add your only the red cells, not your antibodies, okay, just the red cells. If you add the red cells to here, and then they reacted, that tells you you have A on them. Does that answer?
Okay.
Yeah, are you sure?
Yeah, it does. Thank you.
Okay. What about the last one here? All right, what is this? Help me out, please. Okay, I have a few answers in the chat box. Anyone else?
O negative?
O negative, okay. Okay, got a couple private messages telling me. Yeah, I mean, for those of you who participated, it's O negative. Okay, no reaction with antibody A, so no A. No reaction with antibody B, no B. If you don't have A, if you don't have B, that makes you an O. No RH, it's going to be negative. All right. There are a lot more practice for this in the study guide, and you can do it yourself and then check the answers with the in the answer keys. Okay, I'll post the answer key later on. All right, let's oh, I had them typed out. I didn't even realize. Okay, let's try this poll question. Relaunch. Okay, anyone else before I end the poll? Okay. Yeah, Rh negative mother who is carrying a positive fetus, right? That's the only scenario where you would need to have ROGAM. Positive mothers do not need ROGAM. Negative mother with a negative fetus will also not need ROGAM. So if the mother is negative and then they do a blood test on the father and the father is also negative, then ROGAM wouldn't be needed. It's impossible for her to become sensitized, right? My sister is a nurse, and she told me one time about this thing that happened at her workplace. Someone probably made a mistake and ordered a ROGAM shot for an Rh positive mother. Okay, so the mother is positive, and then a ROGAM shot was ordered for her. It was discovered before the shot was given to the mother. So I want to ask you, what happens if you give the ROGAM to a positive mother? Is it going to have no effect on her? Is it going to harm her in any way? Or what do you think? ROGAM shot to a positive mother.
You have agglutination happen
For who? For the mother or for the baby?
for the mother.
For the mother, that's right, okay? Because all her cells are positive, and if you give her ROGAM, the ROGAM will attack her red cells, right? Okay. And then she would probably, it's not going to be too severe, probably. I mean, I don't know. Okay, but logically thinking, it would cause her to have a little bit of hemolysis, right? And it's not going to be a permanent thing because those antibodies will eventually break down. But it's not good for the mother for sure. Okay, so ROGAM is only suitable for negative mother who is carrying a positive baby. Okay, what about this one? Okay, this one is tricky, okay? Please read it carefully and choose carefully. You can choose more than one if you want to. Anyone else? Okay, thank you. Hemolytic disease of the newborn, right? So immediately we can reject A and D because positive mother is not gonna have this problem, right? Not with the Rh blood group anyways. So only B and C would be something to think about. So over here, at first glance, you will see, oh, mother is negative and the baby is positive, right? That looks like it's gonna cause hemolytic disease of a newborn, right? But actually, this is error. The mother is negative, the father is negative. It's impossible for the baby to be positive, right? Okay, so that's not the answer either. Correct answer is C. Okay. There is an other baby matching question. We don't. Yeah, you're right, man. Unless that's not the real father, and then we have bigger issues to worry about other than hemolytic disease of a newborn in that case. There is another matching question here. You can try it out, okay? And if you can't get the answer, or if you need clarification, you can email me. We don't have time to take it up right now. We still have some more slides to go through. All right, let's do this one. Select all the saved transfusions.
Was there an option to choose 2 or?
Yeah, you can choose more than one if you
Okay.
think there is more than one. Anyone else before I take it up? Okay, I think I have the usual 12 respondents. So let's see. A positive to B positive, that is not safe. A cannot be given to B. The B person has anti-A in their plasma, and it will attack and reject the A. O positive cannot be given to A negative because positive blood should not be given to negative. Okay, now I got to clarify something I forgot. Okay? Some of you might say, but you said it's okay to do it once, right? When we do these transfusion questions, we always assume we want to prevent sensitization as well. Okay, so even that one free chance, we're not gonna do it. Okay, so positive cannot be given to negative, ever for these kind of transfusion questions. Okay. C is not safe. AB cannot be given to B. The B has anti-A in them. This one is safe. A can be given to AB and negative can be given to negative. No problem there. A positive to AB negative, that is not safe. Even though A can be given to AB, positive cannot be given to negative. So there was only one answer, which is D. Do we have more? Oh, here is another one. Go ahead. Right. You should not need to do a Punnett square to answer this question. You just have to think about the 4 rules that we wrote down earlier. So we have a person who is AB and a person who's O positive. AB individuals cannot have type O babies. So B and D would not be the answer. Type O people cannot have AB babies. So that's the wrong answer as well. Right? Well, if you cross out 3 is not all of the above. So that's why it's C. If you use the 4 rules, this question should only take you half a minute at most, right? If you do the Punnett square, it's more complicated. Do you understand? Okay, so you want to save some time on the test. How many more? Okay, just one more. Let's do one more and then we're gonna take more notes.
Sorry, a question about the question.
Sure.
It's A negative, yeah? Or
Yes,
just-
A negative. Yeah, A negative.
Oh, Thanks.
If you see a dash after an alphabet on a test, it's gonna be negative. Okay, there's not gonna be like, dashes like that on the test. All right. Okay, so a person who what antibodies could a negative person potentially produce? If you're type A, you can make anti-B. So I see an anti-B here. I see anti-B here, anti-B. And if you are negative, you can produce anti-RH. Right? So the answer is B. Okay. That's it. Anyone have any last minute question about anything that we talked about so far before we move on to the lymphatic system? All right. So we will take more notes. More notes. We will take a note that says, what do we know so far? What do we know so far? Dot, 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 dot. And as well as the lymphatic system. All right. So big diagram, right? Okay, if this is your paper, put it landscape, and then we're gonna start drawing here, and then we're gonna come down, and then we're gonna go back up. It's gonna take the whole page. So what do we know so far? Let's do a quick recap of all the information that we have. Here is our capillary. Okay. And by now, we all know capillaries are leaky. So, let's punch some pores in it. One side of the capillary is connected to arteries. So we call this the arterial end. And this is the venous end. If you think we already draw this, I don't want to draw it again. Draw it again, okay? Because we're going to add things to it that are new and related to the lymphatic system. So what is in our blood? Well, we have, I mean, it's all mixed together in a homogeneous mixture, right? But if you kind of have to separate it and draw them out individually, okay. Like obviously your blood is not yellow, okay, but the yellow portion of your blood, what do we call that? Anybody remember?
Plasma.
Plasma. That's right. This is plasma. Right? It's essentially the fluid portion of blood. Plasma makes up about 55% of your blood, right? And within the plasma, we have the red cells, right? The red cells. Right? We have some white cells as well that I didn't draw here. And then we have some platelets. You know what? Let's draw them. Why not? Okay, so these are platelets. These are red cells. And then we have the white cells, which usually it looks like this. They have multiple nuclei, sorry. Okay, and then we'll draw a circle around it. Okay, so these are WBC, these are RBC, and then these are platelets, right? Nothing new so far. Everything is what you've learned in lecture 2. On the arterial end, if you recall, there is going to be this big red arrow pushing things out, right? That is the blood pressure. And at the same time, we would have a green arrow that's pointing inward. And that represents the osmotic pressure caused by the concentration difference between the inside and the outside, right? So over here, but blood pressure is greater than osmotic pressure. So you end up having fluid loss, right? We talked about that already. So you will have fluid leaking out like this. Not blood. The blood cells don't come out. The proteins don't come out. It's just the fluid that comes out. And when it comes out, right, we're going to have some cells in the tissue. I'm going to draw a bunch of them. Looks like sunny side up eggs. There we go. Just going to draw a lot of them, okay? That's probably enough. When you draw it, you want to draw it all the way to the other side, okay? That's that's the key point. Okay, so these are cells in tissue. Depending on the kind of tissue you're talking about, they could be different kind of cells, okay? Now, these fluid that leaked out, they're gonna, oops, wrong color. They are going to fill the space between the cells. They're gonna fill the space between the cells. Okay, so these fluids that are now existing between the cells, they have a name. They are called interstitial fluid. Okay, so these are fluid between cells. Okay. The fluid portion of the blood is plasma. When it comes out of the capillaries and now exists between the cells, they are called interstitial fluid now. Okay, the composition is very similar, you know, but you're not going to have the proteins, but you will have some dissolved ions, perhaps, right? Maybe you will have some nutrients in them, some waste product, and that's the interstitial fluid. Okay. Let me just double check. I'm actually a horrible speller. I just want to make sure I spell that right. Interstitial. Oh, IAL. Yeah, no wonder it looks wrong. Interstitial. TIAL, not UAL, sorry. Interstitial. Interstitial fluid. Yep. So, on the other side here, what happens to my red arrow? Is it the same size? Is it smaller? What's going on?
Smaller.
That's right, it's smaller. The blood pressure, it's smaller over here. But the green arrow is the same size, right? Osmotic pressure does not change substantially. And so over here, on the venous side, the BP is actually less than the OP. Right? And so you have fluid gain. Right? But as you recall, with the numbers that I gave you, you actually lose more fluid than you are able to bring back in, right? So some will go back in, right? But not all of it. Right? But not all of it. Let me put a star here. Not all of the fluid that came out will be absorbed. Okay. So what happens? What happens to the rest of the fluid? Well, it turns out next to your capillaries, not very far from it, you will always have another type of vessel. These are called lymphatic vessels. Okay, let me draw that one more time. You're gonna have a lymphatic vessel. Okay, that's really close to a capillary. Okay, so these interstitial fluid, some of it will go back on the venous side of the capillary, but some of it is going to go into these lymphatic vessels. Okay, once they are inside the lymphatic vessels, they change their name again. Okay, so these fluid that are inside the lymphatic vessels, okay, they are called lymph, okay, which are fluid inside lymphatic vessels. Okay. Now, just quickly help you see what this relationship is. Okay, like I'm just gonna put it aside. You don't really have to write down. You can just watch. So we have the plasma, which is the fluid portion of the blood. It leaks out, and then it becomes the interstitial. Let me see if I can get it right this time. Interstitial fluid. And then it's going to become the lymph when it goes into the lymphatic vessels. Okay. Then the lymphatic vessels will actually take it back into the blood, and it will once again becomes the plasma. Okay. So those extra bit of fluid that didn't go back in, right? They will return to the blood eventually through the lymphatic vessel. Do you understand that? Unlike the capillaries, which is part of your cardiovascular system, and it's connected to the heart, indirectly, and the heart serves as a driving force to propel the blood, there is no pump in the lymphatic vessels. Okay, the fluid moves slowly, and it relies a little bit on like muscle movement to help circulate them, and so you need to have some valves in there to prevent pooling of the lymph. Okay, so these are valves to prevent backflow. All right, I'm gonna pause there just to see if you have any questions before we complete the diagram.
You know how when the plasma goes into the cells, it becomes interstitial fluid? Does it have a different name when the fluid goes into the lymphatic vessel?
Yeah, lymph, right? Lymph.
Oh, the whole fluid is just called lymph?
That's right. That's right. And they don't go into the cell, right? They go between the cells, right? That's another point. Okay, so maybe I shouldn't have erased that thing. You know, we had the plasma. Once it comes out, it's called interstitial fluid. And then once it goes into the lymphatic vessel, it is called lymph. Eventually, the lymph will go back to the blood, and it will become the plasma again. So essentially, the composition is very similar. You know, just minus the proteins. All right. Now you might be thinking, why go through all this trouble? You know, what's the point of having the fluid comes out and then going into between the cells and then going into the lymph and then eventually just go back to the plasma? Like, does this serve any purpose? It does. Okay, so let's say, and you know, you don't have to ruin your diagram like what I'm about to do. You can just watch. Okay, like I can do it because I can erase it easily. Okay, let's say you have my favorite kind of injury to talk about as an example. Like, you have a gigantic thumbtack that goes into your tissue. Okay, and unfortunately, this little thumbtack has maybe some bacteria in it. Okay, so now there's some bacteria in your tissue. Okay, so what this does is the interstitial fluid might pick up some of the bacteria. It might pick up some of the products that are produced by the bacteria. Sometimes bacteria, they pump out bacterial proteins and stuff like that. And those things would then be carried into your lymphatic vessels. Not necessarily the whole bacteria, okay, but then like parts of the bacteria, maybe, like I said, bacterial products, right? Okay, and you might know this, you might not, but I'm gonna tell you. Let me just choose the right color here. It's not just one lymphatic vessels, right? And you might want to draw this on your diagram. You might have another lymphatic vessel coming from another place. You might have another lymphatic vessel coming from another place. These lymphatic vessels tend to converge, come together, and form like a big knob in your body. Anybody know what I'm talking about here? Like, what is that big knob that that I just drew? You probably heard about it before. Like, you can feel it. You have a lot in the neck area. Your doctor might feel that when you go see them. What is that?
Tonsil?
Yeah, lymph node. That's right. Not the tonsil. Tonsil. We're gonna get to that in a second. But it's the lymph node. I'm trying to draw here, right? So all these lymphatic vessels from all over the body, they will come together and form this lymph node. And you have, you know, big lymph node clusters at several locations in the body. The ones in the neck, you have some around the armpit area, you have some in the groin area, in your legs, right? And these lymph nodes, they serve as hubs to screen the lymph. Okay, so all these lymph that you've collected from, you know, all the tissues is going to pass through the lymphatic, the lymph node. Okay, and in the lymph node, you're going to have some white blood cells in there. Maybe let's use a different color. You'll have some white blood cells there. Okay, could be some macrophage, could be some neutrophils, and these white cells will screen the lymph. Okay, if the lymph has bacteria in it, in our example, then the white cells would react to them. They would clear them out and serve kind of like as a filter, right? And sometimes the infection is really severe. Then the white cells will start to, you know, divide and increase in more cellular activity, and that would cause the lymph node to swell up, right? And that's what the doctor is feeling, right? When they touch your neck and feel your lymph node. A swollen lymph node usually means you have an infection, and your body is working hard to try to clear it. Okay, so multiple lymphatic vessels going to the lymph node, and eventually, and I know this is like gonna mess up your diagram. This blood vessel is gonna keep on extending, right? Becomes the venules and eventually join with the vena cava. And a lot of these lymphatic vessels, after coming together at the lymph node, they will ultimately join back to your blood vessel. And that's why I said in a diagram, right, the lymph will eventually return back to the blood and become the plasma again, right? So under normal circumstances, if there is no bacteria, this is just like a surveillance tool, right? You screen all the lymph that are coming out, and if nothing's wrong with it, you send them back to the blood. And if there's something in there that shouldn't be there, then the white blood cells will clear all those things, and then it will return the lymph back to your circulation. So that's the relationship between your cardiovascular system and part of the lymphatic system. Okay. Are we okay here? Does my diagram make sense? Do you want me to clarify anything? No? Everybody's happy? Okay, then. That's all I wanted to say about this situation. Let's go back to our diagram here and see what we can learn about the lymphatic system. So in addition to, you see all the lymph nodes here, right? Like all these beans looking things, those are lymph nodes. And sometimes you have big cluster, right? Like I said, around the neck and the armpit and the groin area, and further down the legs. It's kind of like at the back of your leg, just a little bit below your knees. That's where you will have your lymph node there. There are other lymphatic organs and structures, such as the tonsils. I'll show you a picture of that later on. You have the thymus, which is in front of the heart. Don't confuse the thymus with the thyroid. Okay, the thyroid is the one that gets big when you don't have enough iodine, for example. The thymus is lower and it's in front of the heart. I showed you the spleen last class, right? When I told you about recycling the red cells, is on the left side of your body, just underneath your diaphragm and behind your stomach. Your bone marrow also plays a role in the lymphatic system, because the bone marrow is where you will make white blood cells, right? And those white cells are part of your lymphatic system. Okay, so lymphatic system is closely related to the immune system, right? And it's also related to your cardiovascular system, which is why I'm inserting this little section here to connect to the next lecture. Okay. Oh, I already wrote the notes. It's the picture that we just draw. So remember when I was drawing the lymph node, when I was drawing the lymphatic vessels, I told you there are these valves in it, right? And that's because the vessels are not connected to any pump, and circulation is really slow. And you need the valves to prevent the lymph from moving backwards and pooling, right? And so if you damage these valves, and it could happen because of a certain type of parasitic infection, it would cause a severe backlog of the lymph. And if the lymph cannot return to your general circulation, then you would have a lot of interstitial fluid building up, right? Do you know what I mean? Like, let's go back to the picture here, right? If the green fluid, the lymph, is not returning, then the blue fluid, the interstitial fluid, has nowhere to go, and they just keep on accumulating in the tissue and cause the tissue to swell up, right? And in extreme cases, we have what's called lymphedema. Okay, and the limbs, you can see here, the leg becomes super big. All these swollen limbs is essentially the fluid that's building up, right? The local people call this elephantitis in some cases, because it swells up so big it resembles parts of an elephant, and it's extremely painful, and it's debilitating as well, right? Okay, so that's lymphedema. We talk about lymph nodes already, right? The clusters around the body. And the main function is to filter the lymph, the green fluid that's going through it, and if there are pathogens in it, or if there's foreign particles in it, then it will increase cellular activity, causing them to swell up. The swollen lymph node usually will resolve itself after a week or so, but if it remains swollen, you should definitely get it checked out. For females, for example, swollen lymph nodes in the armpit area, in the axillary, that sometimes could be early warning sign of breast cancer, and it's not something to take lightly. Okay, tonsils. Okay. We have 3 pairs of tonsils, and they are basically like a collection of lymphatic tissues that has white cells in it, for example, and they will screen for pathogens. And this is a mid-sagittal view of the head. So like, if you cut the head this way and you look at it from the side, and we can see that there is the adenoid. Oh, where's my pen? There is the adenoid tonsils. Okay, so it's really, really far back. You won't be able to see that. When you do your COVID swab, you stick the what do you call that thing? The Q-tip into the nose, right? So like it goes all the way to here and collect the samples, but your tonsil is like even further back. Okay, you're not going to be able to reach it like that. And that would basically screen for pathogens in the air that we breathe. Okay, that could get swollen when you have a respiratory infection. Right? If you get a cold, if you get the flu, right, COVID, that's the tonsil that gets swollen. You have 2 other ones. The lingual, which is at the back of the tongue. That one's really hard to see. Okay, like you're gonna make yourself gag if you want to flatten your tongue with like a tongue depressor with a popsicle stick and take a look at that one. The one that your doctor usually looks at when they tell you to say they are looking at this one at the back here. Get the palatine tonsils. Okay, so here we go. If you feel like doing this after class, look at the mirror, shine some light into your mouth, right? So this is the hard palate. That's the hard part of the mouth. If you stick your tongue up right now, you hit something hard. That's the hard palate. Further back is a soft palate. There's no bones in it. And then there's this thing that dangles at the back of the throat, right? That's the uvula. We'll talk about that later on. But basically, it closes the there's a hole that goes from your mouth to your nose, so it closes that when you swallow it. You ever think back when you were younger? Ever hanging out with your friends and then you guys are eating and then someone tells something really funny and you try to swallow and laugh at the same time, then food end up coming out of your nose, right? That's because when you're laughing and try to swallow at the same time, the uvula doesn't close the hole completely, and the food might end up going backwards. And on the side, then you would have your tonsils. Okay, so these are pretty good-looking tonsils. But when you have an infection, they could turn, red, sometimes orangey, with some white stuff on it. That's when you have a really nasty infection. Okay, so the tongue is right here, and the lingual, it's like you really gotta go in a little bit further to see it, right? You won't be able to. Like I said, you're probably gonna gag if you want to see the lingual tonsil. All right, so 3 of them. The palatine, that's the one we're looking at right now. The lingual that we cannot see at the base of a tongue, and the one up your nose, which is the adenoid. Okay, adenoid. All these 3.
So what causes tonsillitis?
Yeah, so litis is just inflammation, right? Basically, any kind of throat or nose infection, it's gonna cause the tonsils to be inflamed, right? If people have like strep throat, and that's something that's circulating, they're saying kids have a lot of strep A, right now. That will cause the tonsils to become swollen, and they say you have tonsillitis. It's usually not a thing on its own. Is usually a manifestation, a side effect of some other infection that you have, if that makes any sense.
So the infection could be so bad that they have to remove your tonsils?
Oh, removing the tonsil is an entirely different story. Some people have these rocks, okay? They call it the tonsil stones that actually grows in the tonsils, and it causes them to become inflamed chronically. Okay, and in those cases, they might have to remove the tonsils. And you know, it's fine to remove the tonsils. Like you would have a little bit of a little bit dampened immune system, but you know, for the most part, it's not gonna shut down your immune system completely. I think that might be what you're referring to. Okay. The spleen, right? That's like I said, on the upper left quadrant of your body, and it's kind of tucked away behind your stomach. It's in other lymphatic organs, and by now you kind of probably noticed the theme, right? Lymphatic organs, they have white cells in them. They will screen the lymph and make sure that, you know, there's no pathogens in them. That's essentially what the spleen does. The spleen also has a little bit of an extra function. It's able to store a little bit of blood for you. It acts as a reservoir of blood. So, you know, if you are losing blood, if you are injured and you're losing blood, the spleen can temporarily, you know, squeeze out a little bit of blood reserve for you to help maintain the blood pressure. But, you know, it's not a lot of blood. Okay, so like it's not going to be able to sustain you long term. You still need medical attention, but you know, hopefully it will maintain your blood pressure long enough for help to come. It's also possible to live without a spleen. Someone might have to do a splenectomy for various reasons, and you know, after removing the spleen, you will be more susceptible to getting infections. And this is the last slide. Okay, the thymus, right here in front of the heart, is a key organ, as you will see, for producing these not producing, sorry, for so like you remember the T cells and the B cells, right? So they came from the bone marrow. The T cells will leave the bone marrow and it will go to the thymus. Okay, and in the thymus, that's where they will continue to mature. And then when they're fully mature, they will come out and become part of your white cells. But before the T cells can come out of the thymus, they have to take a test. Okay, and only 5% will pass. It's a very strict test. The test tests for autoreactivity. Okay, we're gonna go into more details next class regarding this concept. But every T cells is actually unique. Okay, they have different receptors on them. I'll explain what that means later. But some of these receptors, by chance, might end up recognizing your own antigens. Okay, we talk about how you don't want to be making antibodies that would attack your own cells, right? So similarly, we don't want to be making T cells that would end up attacking your own cells, right? That could result in autoimmune diseases, right? Have you guys heard of autoimmune disease? Anybody have any examples of autoimmune diseases? I'll give you one.
Rheumatoid arthritis.
Yeah, rheumatoid arthritis, right? Exactly. That's when your T cells attacks the tissue in your joints, and it's extremely painful, right? Another one that you might have heard of is ALS, right? That's what Stephen Hawking has. Multiple sclerosis. So all these things are auto-reactive T cells in many cases that, you know, they should have been destroyed and not be allowed to come out of the thymus, but somehow they did make their way out, and it causes a lot of the problem. Okay, so only 5% of them will pass the test. In other words, of all the T cells you make, 5 of them will not be attacking your own cells. And those are the ones that we want, right? We want the T cells that will attack foreign antigen, not your own antigen, okay? And it is also an endocrine gland, which means it secretes this hormone to help with the T cell maturation process. And the hormone that it secretes is something called thymosin. That is all for lecture number 2. There was a lot of information in this lecture, a lot of concepts to study for. So please take the time to go through it. I will post the answer for lecture 2 for you to look at. But I just want to talk briefly about the application questions. Oh, by the way, before I forget, I posted some information on eCentennial regarding our first quiz, right? Let me just pull that up in case you missed it. Quiz 1 will happen next Wednesday, and it will be available from 6 to midnight. 40 minutes. It will only be on lecture 1. 50 multiple choice questions, 4 application questions, and 4 multi-select questions. The multi-select questions, they are worth 2 marks each. You will have to select the correct answer to get full marks. But if you select extra answers that are not supposed to be there, the marks will be deducted. Okay, it's complicated to go through the formula. My point is you don't want to select everything, okay? Because wrong answer will cost you marks. You cannot go below 0 though, okay? So that's not going to be a problem. Is the date wrong for Wednesday? Wednesday, is it the 25th? Let me see. It is the 25th. Okay, thank you. Let me just fix that right now. Thank you. There we go. Okay. I'm sure you're familiar with how to do online quiz by now. But if you're not sure, if you need help or anything, let me know. If you need extra time because you are registered with CALS, please send me the document and then I will accommodate you accordingly. Okay. So, application questions. Bone marrow transplantation can be used to treat leukemia. Leukemia is essentially uncontrolled cell division, often involving white cells. Okay. And so replacing the bone marrow will help it, right? Because you're replacing the stem cells, right? So speculated effects of bone marrow transplantation on a person's blood type. Okay, sorry, Shelley has a question. Do we have lecture on quiz day? Yes, we absolutely have lectures on quiz day. Okay, test day, you don't have to worry about it. Okay, there's no lecture on test day, but quiz you still have to come. Okay, if you replace someone's bone marrow, there is a chance that their blood type will change. Okay, I mean it depends what kind of transplantation you perform, right? So bone marrow transplantation follows the same rule as blood transfusion. Let's say a person is type B blood. So for bone marrow transplantation, you can either give them type B donor marrow or type O. So if you give them type B type B bone marrow, nothing will change afterwards. But if you give them type O bone marrow, then it's possible for the person to change the blood type from B to O, right? Because the new stem cells will only be able to produce type O cells afterwards, right? And that could change the person's blood type afterwards completely. I actually had a student when I taught this. She was telling me when she was a kid, she actually had leukemia. And after doing bone marrow transplantation, that's exactly what happened to her blood type. It changed. Okay? I forgot what it was, but it changed from maybe from B to O, like in this case. Okay, so that's possible. Number 2, Sally's first child was born with hemolytic disease of the newborn. She learned about RhoGAM from her friend. Sally is now pregnant with her second child, and she is requesting to receive RhoGAM shot from her doctor. Is Sally's request valid? What do you think? What if you become the nurse and then someone like Sally is asking you to give her RhoGAM in this scenario?
No, because she's already been I forgot the word, not sterilized.
Sensitized, right? Yeah.
Sensitized, yeah.
Yeah, you're absolutely correct. Okay, RhoGAM will not help her because she already got sensitized in the first pregnancy. She already has the anti-Rh antibody, right? RhoGAM is to prevent sensitization, right? So if you give her RhoGAM now, it would not help with the second child. Okay, now you might wonder, does that mean people who are sensitized cannot have second babies? No, not necessarily. I mean, there are other interventions that could happen to help prevent hemolytic disease of the newborn, even though the person is sensitized. But there are greater risks associated with it, right? For example, one thing that they could do is if they discover that the baby is being attacked by the antibodies, midway during pregnancy, sometimes they would do in utero blood transfusion. They would do transfusion for the baby with the baby still inside the mother. That's possible to help with the situation, right? Because a lot of the red cells are dying, so it makes sense to give the baby some new blood, right, to help fix the problem. So that's one thing that they could do, and there are other interventions that might be possible. But it's all about making an informed choice, right? So if someone already had sensitization history, right, they should know what are the consequences if they choose to have another child. Now, there is another, subtle hidden thing in this question that you might have missed. Okay, let's read the first sentence again. Sally's first child was born with hemolytic disease of the newborn. How exactly would that happen, though, right? Based on what I explained to you, it was the second baby that could have had a problem, right? But how come in this case, the first child is experiencing hemolytic disease of a newborn? What's going on here?
Sally was sensitized before her first pregnancy.
Exactly, right? So pregnancy is not the only way to get sensitized, right? Blood transfusion can cause sensitization as well, right? So maybe Sally once in the past received a positive blood transfusion, right? And then she was sensitized, and now those antibodies cross the placenta to attack the first baby. Okay. All right, so that is it. We will do lecture 3 next week. Each unit I try to space out the difficult lectures throughout the course. So each unit has one very complicated, difficult lecture. And lecture 3 is that one for this unit. Okay, so make sure you come to class, bring your notes, and I'll try my best to explain it to you. All right. Have a fantastic week. I'll see you all next Monday. Bye for now.